Welcome to the third and last webinar organized by the EUFORGEN program related to the Ash Dieback and its impact to the ash genetic diversity. So EUFORGEN, the European Forest Genetic Resource Program, is a cooperation program on the conservation and sustainable use of forest genetic resources in Europe. EUFORGEN is an implementation mechanism of the Forest Euro process, the high-level forest policy process that aims to develop common strategies on how to protect and sustainably manage European forests. Today, we have two guest speakers who have kindly agreed to share the knowledge and recent scientific findings on the natural selection of ash population related to the spreading of Ash Dieback. The first speaker, Erik Dahl Kjaer, from the Department of Geoscience and Natural Resource Management from the University of Copenhagen, will give a talk on implication for wise genetic management of natural ash forests and genetic conservation areas infected by the Ash Dieback. Erik, the floor is yours.
Can you hear me?
Yes, we can. We are stopping to share the screen now, and then you will be able to share yours. Perfect.
Can you see my screen?
Yes.
Okay. I'll start by thanking you for inviting me to talk on this very important topic that that I've worked for some years now. Natural selection ash population related to spreading of Ash Dieback. And here today we'll focus on what happened in the mature forest and mature plantation and old growth forest, also relation to conservation.
Erik, we cannot hear you. Erik?
Can you hear me now?
Now we can, yes.
Okay. Okay. Yeah,
Yes,
so the
okay.
point of the departure is that we have observed that there's a lot of genetic variation in resistance or tolerance to towards Ash Dieback. And that of course opened the questions for how can we then utilize that in a smart way in genetic management to preserve the ash trees and ash forest. We found out quite early when the Ash Dieback hit Europe that this was the case. We have the variation. And here I show some pictures from a seedling seed orchard. So that is like a progeny trial consisting of 97 families. That is located in Denmark, and this is a site where was planted in 2002, and we think that the disease hit Denmark about that time. So we see here in 2009 on the top left, you may see a picture of there some most of the trees are dead, but there are actually some trees which is alive and fairly healthy. And this is a family plot, so the trees that we see next to Levi McKinney, a PhD student that time, all belong to the same family. And below you can see how things looked 5 years later. It was a clear effect of some families were performing much better than others, and based on that we could estimate genetic parameters and heritability. And you can see 2016 that already by then that some of the trees has grown quite big and were at that time fruiting. So the point here would be that we have genetic variation that we can use forward. This was then actually developed into a first generation seed orchard now. So it's a seed source where people can actually get seed. Because we could estimate heritability and genetic parameters, that means we could have an expected gain here. So the gain will be an effect of the selection, how intense you select, and the heritability. And from there, we could select seeds, like from the tree behind my colleague Lars Nørgaard. You can see we collect seed from the trees there, and then we could make start for a second generation seed orchard. And this is a project in cooperation with Nordic partners, supported by SNS, together with the Nature Agency. We could, from 80 trees, we could select seed and then sow the seed and expose them to disease and strong forward selection. So this is the spring last 2021, where we can see here seedlings being infected, and then we could do selection, and We could transplant them into the nursery where they could still be affected more, and then we could do sorting, and we could then establish here the second generation seed orchard in cooperation with the Senate. This was planted last year, and based on that, then we have the idea we could do breeding for having better seed sources, and that's the last seminar was discussing about activities in other countries. So this was, of course, very encouraging that we seems to have a tool here that we could find good seed sources. Now, what about all the old plantation and this old growth forest? I mean, it is not really a very positive solution if that would require that we have to remove all the old ash forests and replant them again. So what I talk about today is actually what will happen in these forests and what can we do about that. And this is something that we from very beginning was focusing on. You know, what can we, how can we, what will happen in these forests? And there was a number of questions that we thought about that time. So the first, I mean, there will be selection going on also in this old plantation and old growth forest. But what will happen? I mean, when will they die, and will they all die? I mean, hopefully they will not all die. But there's issue about how soon can we see which will be surviving and will not survive. And if they will survive for a long time but be high susceptible, how much will they contribute to next generation? So that would be the next question about generation turnover. I mean, who would actually sire the next generation? Will it be the unhealthy trees that start to fruit and flower heavily and they spread a lot of seed and pollen? Or will it be more the healthy trees that, due to their better vigor, actually will be the main parents? Then another concern would be if we had this case where only a few trees will survive and many trees will die. I mean, what would be the implication for the genetic resource of these populations? Will we create a genetic bottleneck leading to lost diversity? Or even though we keep diversity on the landscape level, will there be local inbreeding? Because in a given forest or population, the next generation will be sired by very few trees. Another important question that we have been worried about, what kind of tolerance, I mean, are we dealing with? Will this tolerance last, or will we see that within 10, 15 years, what used to be tolerant will now become susceptible? There were other questions. Think about the ash trees and the ash forest and as an ecosystem rather than a tree. There were the microbiomes. There's also actually how to do active management for improved tolerance. I mean, should we make enrichment plantings? What kind of silviculture? What would be the ecology and dynamics with other species? I put them in shade here because I'm not have no time to cover that today. So I keep a focus on the first one, but maybe a few words on the last one also. So the first about selection. When will they die and will they all die? When the disease hit Denmark, we set up some service stands. This has been done in different countries, and Denmark we set up 8 service stands, and we've been following since 2010. Each of these stands have 50 individuals that we've been following. The results was recently published in a paper as part of Corey Lyne Madsens PhD work, but we of course still follow these populations. These stands have known origin, so we know exactly where they came from. So we know it's the same origin. They came from seed sources or approved seed stands, and they were different ages. We selected some were quite young, must have been about 10 years when the disease hit, and some was old, more than 90 years when the disease hit. I'll just show you some results. Here we have here we can see different colors will represent the trees with different kind of disease level. So in heavy green, you will see the portion of trees which were completely healthy. There's no symptoms. In light green, that would be trees that have some symptoms, but we still consider them to be healthy. Then getting to more and more susceptible, and the black one is the ones that died during the survey. We have 2 populations here, one which was 60 years old in 2022, and one which is 65 years old. So they must have been 40 to 45 years old when the disease hit, and they came from the same origin, the same seed source. So 2 observations. One observation is that the results are very different. So there's a very strong side effect here. On the one side, in Alice, even today, we have about 20% of the trees that actually look quite healthy. While in the others that have the same genetic origin, we see very few very healthy trees by now. We also, another observation is that there's a decrease, even though 20 years after the disease hit, we still see a negative development. We still see trees die. So we're not really at the end of observing what happened. The third observation could be that actually mortality started quite late. When we started assessing these things in 2010, I had this expectation that the trees will die very, very fast, but they didn't happen. You can see many trees have died, but it took some time before mortality really hit. They were very unhealthy, but some of the really unhealthy trees that was really unhealthy, even by then, are actually still about to die. Now, this is what we call a mature stands. We have then the younger ones, and this is just two examples. The one from Haderslev and one from Stenderup, which at that time was 10 and 20 years old. So they were very young plantations. And you can see here, that's been shown in many other studies also that the young trees are actually subject to very high mortality. And I should say that we started the survey in 2010, and for that time, at that time already there's been high mortality in these young plantations. So I suppose maybe half or maybe 30% of the trees was already dead. So what we see here is maybe only half of the mortality because many trees died before the survey. But on optimistic note, we can see actually even today there still is a fraction of these trees that are actually quite healthy. 10% or so, and if we assume that half of them were dead already when we started, that would leave us still with 5% that looks really healthy even after all these years, and even in a situation where many trees have died. So obviously there must be some selection going on here that could lead to gain, because we know that we have genetic control behind this trait. Well, and then again, maybe we do not know, because I mean the data we have on heritabilities and various components are from progeny trials established on previous agricultural land, planted carefully, measuring, and in real life, of course, things are very different. So we don't really know what will happen in these natural populations. On the one hand, here the selection can be very, very strong because there could be a huge number of seedlings per hectare in such a site. On the other hand, the heritability can be much lower because it's a much more heterogeneous environment. There has been at least one attempt to try to estimate this in situ heritability, but it's quite complicated, and we're working on that also. My colleague Lene Rosgaard Nielsen also have a project trying to understand that. Another a way to try to understand what's happening is actually to use genetic markers to do that, and that's also something that we are working with in our group. But later, Richard will say something about that, so just skip that. But just to say that there is a lot of potential for selection, but I think really today we're not really sure how that will go to work, because we still need to have a handle on heritability, and that's something that we'll work on. Okay, I have to move on. So next question would be on the what we can conclude on that. So I think conclusion could be that mortality is very fast young plantation, but much lower old populations. Mortality will increase even after 20 years of the first. So the trees are still dying. There's large variation among sites. There's also a very important effect of secondary infections. I think Michele Bozzano talked about that, and I will not say anything, but it's also part of our study. We try to see the role of secondary infections and others. And on the positive side, there seems also in this forest to be something like 5% or something that is actually seems to be remain tolerant even after very heavy infection. So I move to the next question: what will happen at the generation turnover? This is the work by PhD work by Devin Cummings, Devin that maybe is online actually. So basically the question here was, will be the unhealthy trees that flower heavily, or will be like the healthy trees that have the highest reproductive fitness? And there's also a gender issue here, because we have male and female trees, and will there be some difference there? It's based on 3 papers that Devin has published as part of a PhD, and I just show a few results. The first result is based on actually study in the clone seed orchard. This is a case where we have 39 genotypes, clones, which has been studied very carefully. So we know a lot about these clones, and we know about the susceptibility, we know about the gender. And then we've been, Devlin was studying the reproductive success. And here you see on the left hand, you can see on the top panel is the female part, so it's the seed production. And on the x-axis, you have how susceptible this tree is, and on the response is how success in terms of seed set. And on the picture, you have this tree number 35 with one clone, and you can see that the green arrow is actually very, very healthy, and it's also very productive. So there's a clear correlation here. The more susceptible the trees are, the less they contribute to next generation. On the male side, it's not so completely clear, but this is based on looking up based on genetic markers to find out who's been siring the offspring, and we can see here again we have a very at the very left we have our most healthy male clone is actually also the most effective pollinator, but it seems to be less clear on the male side, which may make sense because actually you need some quite an investment to make a seed where actually pollination could be easier for an unhealthy tree to produce some pollen before it will die. This was under control conditions. So what will happen in the old forest that we're discussing today? And Devlin also looked at that in two studies, one in Denmark, one in Germany. I'll only show results from the Danish study. So we have here it's a forest with the ash planted, and what you see here in the pie charts are the distribution of trees in terms of health classes. So the dead ones are the ones which was very recently died, and the green ones are the ones which are healthy, and then the other colors would then indicate an increasing number of unhealthy. At the top you have the ones which are mainly male trees, and on the bottom the ones which are mainly female trees. And you can see the contribution is almost the same between male and female trees when it comes to level of infection. Now then on the right you can see the pie chart showing who have been siring the seedlings. So Devlin made some transects through the forest and some seedlings, and here you can see who have been siring the seedlings. And there's a picture here that is actually mainly the healthy trees that are, I mean, they're overrepresented in as parents. So while there were maybe 15% healthy male trees out there, it is certainly more than 25% of the male parents that came from health that are healthy, and a similar picture for the females. So we can see that that actually that it is definitely the healthy trees that produce more offspring, and that is of course very good news. So you can see healthy trees are overrepresented as parents for next generation. Frequency of healthy trees are low, and unhealthy trees may therefore still size substantial amount for next generation. This probably will lead to a positive selection in the generation turnover, since healthy trees produce more seed, and since selection during the growth of the juvenile seedlings will most likely also promote healthy trees. How fast it will work and the long-term effect, we still have to work on that, but it's good news anyway. Now, then going to effect of bottlenecks. What can we expect bottlenecks will be developed during to this, that only a few % of the trees will survive and be heavy seed producing. Again, this is something that Devlin studied in her studies both in Denmark and Germany. I just show results here from Val-Behind Forest. This is this dispersal curve, seed dispersal, and pollen dispersal curves. Yeah. So at the x-axis you have the distance, and on the y-axis you have the cumulative probability of dispersal. If we say like take 90%, we can see that actually 10% of the seed are dispersed actually more than 100 meters from the mother tree. When it comes to pollen, it's very long distance. A lot of 10% is dispersed more than 800 meters. So even though the seed is a bit heavy seed, then we have observed that that can be spread quite long from the mother tree, and I think that's probably has to do with the fact that the seed is very often sitting on the trees until you have a very strong storm, and then the seed can flow. Devlin also looked a lot about details about seed dispersed direction and so on, but that you can find in the paper. The conclusion would be that a single healthy tree can actually spread seed over a large area. So a very productive trees could actually cover seed for 3-4 hectares. So one tree per 3-4 hectares would actually be able to spread seed, and pollen will actually connect also fragment populations. In another study that Devlin made, she actually looked at a fragmented populations in Germany and found also a very long, very clear dispersal of pollen between populations. When it comes to loss of diversity, it seems like this is a less problem because of this effective dispersal. But we have to be careful about correlate response, and I'll not talk about that. But if we do this kind of strong forward selection for one trait, and this trait is correlated basically, one can think about planting tolerant trees from breeding programs in the landscape, and then can spread the genes and the seed into old growth forest, and therefore improve the general level of tolerance in this forest. So the last topic I'll cover is about the tolerance, whether it be lost or not. And that's of course a big concern. I mean, what will happen when we fill the forest with the tolerant trees? Will the fungus then evolve and become much more pathogenic? So the trees must grow for a long time, while the pathogen actually is a sexual recombining pathogen which has 1 year generation time. Okay, so just go back to what Michele already told about this the biology of this fungus. This is I took this from a very recent review made by McCann and others in forest microbiology. So the spores are being produced on the apothecia, on the old petioles in the forest bed. Then it will be fly into and infect the leaves. It goes be in the leaves, and when the petioles fall down next year, the fruiting bodies will develop, and then it will spread again. So this is like the cycle of the fungus. It, as we know, also have this very unpleasant feature that will also go into the tree and damage the trees and kill the trees. Now, the point here is that actually the whole fitness is not really related to whether it's a pathogen or not. The whole fitness is related to ability to invade these leaves and the spot and infect trees, or not in terms of being pathogen, but only if they can infect the leaves and produce spores on the old apothecia. So that would be then the hypothesis would be that even though we introduce tolerant trees all over, this will not trigger a selection for pathogenity. So this is something we looked at, and this is the work by Cheta Kusavang. The idea would be to go out there and sample in the landscapes, in the forest, sample different isolates and try to infect trees, and then see if some of these isolates were actually non-pathogenic, even though they would be present in the forest. So this is just the results. It is here in the x-axis you see day after inoculations, and on the y-axis you see how the necrosis developed for different isolates, and there's very big difference. I mean, there are some highly pathogenic isolates, but there was also some which much less, and actually one which is completely non-pathogenic. This is called 22.1. And if we look at it, it's look at how it grows in vitro. It's actually grow very fast. It actually so happens that here on the x-axis you have the in vitro growth, and on the y-axis you have the pathogenity. And this 22.1 actually was the fastest growing in vitro, but without having any apparently making any symptoms on the tree. I mean, there are many details that you can find in the paper. So, I mean, the biology of the species suggests that the pathogen may not be related, the pathogenicity may not be related to fitness. Most isolates in nature have probably never functioned as pathogen. There's also some studies indicating that, and that support that the support from this study, at least, that there is presence of these non-pathogenic isolates, which means some evolutionary optimism. I mean, maybe we can just maybe the fungus will just turn out from being an aggressive pathogen to being an endophyte, as it is where it comes from. Okay, this was the topic I covered. We could say something about ash forest and ecosystem, and hollow ponds is very important. Hypothesis that the hollow pond could be an important part of the tolerance. That's something we should look more into. There's different Chetak Kasawang and also Fang, our PhD student, is trying to look at that, and other people doing the same. When it comes to silviculture, there's many things to look into. This is just one slide I'll show you. It's based on simulations where we try to simulate using this Silva, which is a forest simulation tool, trying to understand what is the likely development over time if you start up having very few surviving seedlings compared to many. And without going details, the green curve would be what we expect if we just by now, and the other ones would be different kinds of more tolerant seed sources. And the take-home message here would be, if we actually introduce more tolerant seed sources, maybe we could have actually a development in the forest, which is not too much different from what we managed in a managed forest. And so many things to discuss, how to do thinnings, what the role of mixtures is also something we try to look into, the species dynamics, enrichment plantings, whether the short tongue or long tongue. These are things that will be covered also in a new EU project, this Opt Forest that just started last year, where we try to integrate genetics into discussing silviculture. So I'll not say any more about that, just to say that to conclude that we had this question when we started, when it comes to selection, we still have to see what happened, but in general, this seems to be positive. I mean, they will die, but they will not all die. That's what it seems. On generation turnover, we think it's very positive. It seems that it's actually the healthy trees that are next generation. So there'll be ongoing positive selection. When it comes to dig bottlenecks, we think it's optimism. We don't see from this kind of gene flow, we don't expect to be severe bottlenecks. When it comes to tolerance, we see most things indicate towards actually that this is a tolerance that has a genetic background that actually and biological background that make it durable. And then there are other things we need to look more into. So there are many things to discuss. Okay, my time is up. So I think any times for discussions or questions, that would be now.
Thank you very much, Erik. Really very, very informative. I will encourage everyone online to just raise your hand to ask for the floor to ask questions, or you can type it into the chat box. I will just ask you kindly to focus not on the details, because then we could be spending hours speaking about details or technicalities. I've seen that actually James Brown is asking the floor. He has already indicated this question. Please, James, you can unmute yourself and ask the question directly.
Hi, Erik. It's a really great talk. I mean, it fits absolutely with our observations in England. Really pleased by that. I've got two questions, if you don't mind, Michele. One is, Erik, would you expect to see a shift, the result of natural selection being a shift in the range of ash to drier areas? I mean, we see this very clearly here, that it's worse in wetter, waterlogged, deep organic soils and much less in drier locations. The other question is, and it's somewhat related, is do you really mean tolerance? How sure are you that what you're seeing is tolerance rather than resistance to the pathogen?
Yeah. Okay. Thank you, James. The first question, yeah, I agree with you. I mean, these the two populations I showed you, they are different. The one is what's more and more wet and damp area. Unfortunately, in this country, we have a lot of ash on these very wet areas, and there where we see the highest mortality. And it seems to be the fact that the fungus can spread more because it needs this wet area, but also that we have the secondary infections. We see more severe infections of the armillaria. It seems to be outside the forest we see much less symptoms. There's a study, the French study, also showing that that distance from the forest actually also makes difference. So the trees in the landscape are less affected than the trees in the forest. The worst affected is the one in the very wet area. So I think you have a point there. Again, if we could, if we have get more tolerant trees or resistant trees, that would then of course allow the trees to maintain this very important in our forest. It's a very important ecological niche they take. Now, tolerance resistance. Now, maybe I should leave that to you. I haven't had a long time to find out what to call this, because it's not complete resistance. I mean, it is at least partly resistance. And when I call it tolerance, it's because I mean the pathogen actually infect the leaves. The whole idea is that it does not cause any pathogenic response. But I will leave it to you, forest pathologists, to discuss that more, because. But my point would be, the reason I call it trolls, because the fungus do infect the leaves. We do see that. We do, when we do qPCR, we do find the fungus in the leaves, but it does not develop symptoms. Okay, so that anyway. But I'm not the real writer to go into details on that discussion.
I would say that, I mean, in our own experiments, what we saw was resistance, but we weren't studying many genotypes. And I think my own observations, but we haven't done this rigorously enough, is that we're seeing both, but probably more resistance. But I have seen evidence that there is genuine tolerance. So the path of trees getting infected and then continuing to grow. But I think we also see cases in which trees get infected less than their neighbors. So anyway, I think this is I think the question of resistance or tolerance is interesting to follow up. But Arno has made exactly the point that I was just about to say, is we're really looking at low susceptibility, whether it's resistance or tolerance.
Yeah.
Thanks very much. Thank you.
We do see the fungus in the leaves when we do qPCR.
Yeah.
Thank you very much. Then we have another question for Bertolt Heinze. Bertolt, I don't know if you want to unmute yourself, or I can read the question out loud.
Yes, I'm trying to unmute myself.
Yes,
right.
We can hear you.
Yeah, okay. Thank you. Hi, Erik. Great talk. My question was related to the strains of the fungus being either more or less pathogenic, and what we expect for the future. Wouldn't you, I mean, in the very long run, expect that the less pathogenic strains would take over because they do not kill their hosts?
Yes, I mean that's a good point. It's a good. I agree. So, I mean, there's been some studies. I think one of the challenges is that people work with isolates that are isolated from heavily infected trees. So here, actually, this isolate was actually sampled just by going out in the forest and sample the spores. That was the challenge, and there we find these less pathogenic. And I agree with you. I mean, in at least in the long run, we would expect that this should be the case, that actually it would be actually better fitness to be non-pathogenic than being pathogenic. But I mean, this is one study that worked by Kjaer. It actually took a long time to make it, but I mean, we need to study them into much more into details. But I mean, the results is optimistic because it supports what we would expect from a more evolutionary point of view.
Thanks again.
Thank you very much. Then we have a question from François Lefebvre. François, if you can just ask yourself, otherwise.
Yes. Hello,
Yes.
Erik. Thank you for the talk. Yeah, my question is, do you have any expectation on the possible role of silviculture on the disease dynamics? I raised the question because you showed some picture with two stands with the same genetic background, let's say, with different dynamics. Could it be related to environmental conditions or to different silvicultures? This is.
Yeah. In this case, I think it's environmental conditions because it's probably to do how wet the area is. So, I mean, the cool thing about these conservation stands or service stands is that it's the same genetic background. They came from the same seed source. So we can see, and I think in this case it's environmental. But I think there's a very important role in discussing silviculture. And I'm asked again by foresters, how should we do it? And together, in this forest, as you know, François, together we will actually look at that. And I agree with what forest cooperation to working the more advanced modeling of silviculture. I think that's the way forward. Now we have a lot of big parameters coming into the system. We need to do some estimate in situ heritability, and then I think we can come along with quite advanced modeling that is platforms are being developed in France. So I think I don't know, but I think there'll be a role silviculture. That's something that I think is been farly understudied so far. Everybody focused on the genetics. We have to look more on that.
Thank you very much. Actually, I see that there are really many other questions, but I'm trying to limit. But we can still have another discussion at the end of the other talk. I will have a question myself now, Erik. What happened with Ash? It was dramatic in many cases, but for us, it's also an important learning experience. we can expect that in the future there will be many other cases of new pathogens coming into our landscapes. So I don't know if you will have any lesson learned from what has happened to the ash to be more prepared in case something will happen for other species. What should we have in order to be able to cope with, either from the scientific point of view or from the practicality, the protocols that we should be following?
Yeah. So first, thank you for this question. I mean, first point would be, I mean, when this disease hit, I think that we had a huge advantage by cooperation amongst research groups, amongst countries. And I mean, I'm not a forest pathologist, but you know, working with forest pathologists would allow us to put our expertise together. And internationally, we had a lot of cooperation. I think, you know, that we had a really good handle on this one because many people work together. I think that's something we should really learn. If that happened again, we should do this kind of work. Another thing is that we had this advanced network. We were kind of prepared. I mean, I showed this pictures from this seedling seed orchard was established by the Nature Agency in 2004. Before the disease, actually exactly what the disease hit, but at that time nobody would know. So in Denmark, the Nature Agency has developed this kind of seedling seed orchards for more than 25 different species. And these are very, very important resource because once the disease hit, we can go in and look for genetic variation and estimate parameters and college bonds, but also we can just start very brief at developing seed sources. So I think that's also something that's being prepared, having the technologies, having the cooperation, but also having these things in the field already, I think is very important. Then also we could learn things that we should have been more aware about. I mean, many things. I mean, we were too slow to respond. I mean, one of these things that we work with also sentient gardens where we try to look, you know, if I've taken on vacation to Tokyo or China and went to the botanical garden and I would say, oh, how come that European ash trees look so unhealthy here? Then I would have maybe realized that there was a problem coming up. So this is also this kind of international cooperation that we should try to think about that, you know, be prepared and there's something about early detection. There are many things that we should work on. Yeah, that was a very long and that's very precise answer to a short question.
Thank you very much, Erik. Really very, very important for us. A lot of food for thought, and we will be building on this in the next days and years to come. I would like now to switch to the next presentation from Richard Baggs from Kew Royal Botanical Garden and Queen Mary University of London that will give a specific example about genomic evidence of a natural selection by Ash Dieback in an unmanaged woodland. And then after your presentation, Richard, we will go back both to questions specific to you and maybe we will have a few more minutes for a broader discussion again. Richard, please, the floor is yours.
Thank you very much, Michele, and thank you to you and your colleagues for organising these webinars. They've been really, really useful, and I've just learned a lot from what Erik has said, so thank you very much. Well, what I'm about to say complements very much what Erik has just shared with us. We've been looking at natural selection in an unmanaged woodland in Britain. At the webinar last week, I talked about our work on the genomic basis of ash resistance to Ash Dieback and how we could get 67% accuracy of assignment using genomic prediction based on 10,000 SNPs. And that was based on planted trials. And so, as Erik says, there's quite a difference between planted trials and a natural woodland. And back in 2020, when we published a review in Plants, People, Planet, we said in most cases, if a breeding program is viable, then natural selection is also likely to be effective. The decision to embark on a breeding program may therefore often be dependent on an assessment of the relative speed at which allele frequencies can be changed by a breeding program versus natural selection. And since I wrote that, I've sort of gone back and forth a little bit about how true that is. And Erik introduced us to the breeder's equation, which is very familiar to all of you. I'll just bring it in by means of introduction. And of course, most of you know more about this than I do. The change in a mean value of a trait in a population in one generation is equal to the selection differential, the strength of selection, multiplied by what we call the heritability. And that is the proportion of the variation in the phenotype that is due to genetics. And of course, this involves also looking at the amount of phenotypic variation due to the environment. So when we first started to think about this, we thought, well, we've got high predicted mortality, so the selection differential should be high. We know that the Ash genome is pretty variable in Europe, and from studies, most notably by Erik, we knew that heritability of low damage in family and clonal trials was around 0.4. And so things look pretty good from those numbers in terms of a response to selection, or indeed natural selection. But we do have to bear in mind, I'm just repeating Erik now, actually, we're saying exactly the same thing. In a woodland, things are very different to in a planted trial. Not only do you have different tree ages, but you have larger environmental variance due to the different spacing, the different shading of the trees, competition with sometimes with different species, mycorrhizae, herbivores. We have lots of deer in the UK especially, and other diseases like cankers. And so all of this is going to make environmental variance much greater in a woodland setting. And I'm just noting here, in the UK, most ash woodland is unmanaged, or if it is managed, it's managed for pheasants rather than for trees. And we also had to think about the effect of the environment on the fungus as well. We've discussed that, how it likes wetter environments. And so there's another way in which the environment gets involved as well. And so in a woodland, we expect lots of environmental variance, which means a low heritability. And if there's low heritability, change might be really small and really slow. So we studied this using our molecular markers. We haven't managed to publish it yet, but it's up on bioRxiv, and do read the paper if you want to follow up on anything I'm saying. And the PhD student who did most of the work here was Carey Metheringham, and Richard Nichols, a professor at Queen Mary of University of London, was designed the statistics, and they got quite complicated, as you'll see, and it was all his expertise. So we studied a woodland called Marden Park Wood, managed by the Woodland Trust. It's found just to the south of London, and here is a very detailed map of it. Tree size is shown by the circumference of these circles, and so you can see we've got a lot of different tree sizes and a very healthy natural regeneration. This is what the canopy looked like. So we went through and we scored the health of the trees. With the adult trees, we just had to stand on the ground and estimate canopy loss. It's not an ideal method. We've tried using drones, but at the moment we're just relying on ground surveys. And then for the natural regeneration, we went through and scored the trees using a similar scoring system as we used in the planted trials that we'd worked on in our Stocks et al. Paper. Here's two students doing measurements. And just as Erik was saying, we found much faster disease progression in the juveniles than in the adults. We think that when we started the study, no adults had disappeared from the population due to Ash Dieback. Some might have been still standing and dead, but none had disappeared completely. But with the juveniles, of course, because they die much more quickly, we expect that many juveniles had already died, and we could not detect them because they had disappeared into the undergrowth. So we did whole genome sequencing on 133 adult trees, which were established before the pandemic, and 442 juvenile trees, which we think had established since the epidemic arrived. And we looked for the top 10,000 GWAS SNPs that we'd found in the Stocks et al. Paper. And interestingly, 10.6 of them were fixed for the good allele. Trees only had the good allele in the population. 80% were variable, and 9.6% were fixed for the bad allele, the allele associated with high damage. So this is quite promising because 90.4% of the alleles we want are already present in the population. Now, the first thing we did was calculate a genomically estimated breeding value for each individual and had a look at whether that correlated with our damage scores, and they didn't, basically. Now, you might say, well, that's because your GEBVs are just inaccurate. But the fact is, as I mentioned before, environmental variation is going to be so great in the population that a lot of the damage scores are going to be affected by the environment. So we would not necessarily expect them to correlate well. And you also have to remember that the most susceptible juveniles will be a missing fraction. They will already have disappeared from the population without trace. They might have been killed by the disease before they even laid down woody tissue. So what we then did was we compared the mean genomically estimated breeding values of the adults versus the juveniles. And we found that the mean values were 7% higher for the juveniles. So there's this slight shift. So that was what we were hoping to see. So that suggests that the juveniles are more resistant, that on average, genetically, than the adults. And comparing just the two means, statistically, this was not significant. But we have to bear in mind, a lot of these trees are very related, and we are just comparing two means. What we then did, and this is quite complicated, but allowed a really good insight, is rather than comparing the juveniles with the adults, we took a different set of SNPs, about 6,000 neutral SNPs, spread across the genomes, completely different set of SNPs to what we'd used before. And using the genotypes of the adults and the genomically estimated breeding values of the adults, we trained a new genomic prediction model based on these neutral loci. And we then used that to predict genomically estimated breeding values of each juvenile. And so that gave us a GEBV that was based solely on their relatedness to the adults in the population. And then we compared that GEBV to the GEBV based on our 8,000 SNPs from the Stocks et al. Paper. So what that meant was we compared the breeding value we'd expect without natural selection with the actual breeding value that we found. And we found this shift, but now it was just 5.8%. It was slightly smaller, but we had a lot more power in our statistics now because it was effectively a pairwise analysis. Each individual had two values that we were comparing, and this was highly significant. Now, this shift of 5.8%, that excludes the effect of differential reproduction by the adult at the seed and pollen production phase. Because for each individual, we've calculated its relationship with the adult. So this shift, we think, is mainly due to actual death of juvenile trees. So we have a significant shift. And so that enables us to calculate how much natural selection is happening. Now, unfortunately, as Erik says, we don't know what the heritability is in the wild. It could be 0.4 like in the trials, but we don't think it is. It could be as low as 0.1. We just don't know. And unfortunately, we couldn't use our data to calculate the heritability because of the relatedness structure within our population. All of the methods that use genome-wide SNP data for calculating heritability rely on unrelated samples, so we couldn't use those methods, unfortunately. But if heritability is, for example, 0.1, that would mean there's 60% elimination by selection in the juvenile phase. Another thing we did was for some of the juvenile trees, but not all of them, we could work out their exact parents. Unfortunately, because close relationships, we couldn't do this for all of them, but for some of them we could. And for those that we could, we compared the phenotype score of the offspring, which is here on this vertical axis, first of all with our canopy cover measure of the phenotype of the parent, and then also with the GEBV of the parent. And you can see we found no relationship between the phenotypic score of the parent and the phenotypic score of the offspring. But we did find a relationship between the GEBV of the parents and the phenotypic score of the offspring. So it suggests that GEBVs would be a better way of selecting parents for a breeding program than merely relying on phenotypes. Now, of course, this is in a woodland situation where we're looking at continuous variability within a population. I'm not saying that going out into the wild and finding a population where all of the adult trees are dead, but one of them is looking healthy, we're not saying that wouldn't work, but we're saying in this situation where we're comparing trees with a continuum of phenotype scores, that these are the results we got. So in conclusion, a single woodland patch contains 90% of the alleles, the 10,000 alleles that we wanted for ADB resistance. GBVs were not good predictors of ADB damaged phenotypes, but this is expected as the phenotypes are so affected by the environment. We found a shift in GBVs in the juvenile trees attributable to natural selection, and parent GBVs were better than parent phenotypes at predicting offspring phenotypes. But we have lots of questions we haven't answered. Can natural selection save ash? We don't know yet. If an ash tree was fixed for all of the good alleles, all of our 10,000 good alleles, how resistant would it actually be? We don't know. Are ash trees outside of woodlands under similar levels of selection? We don't know. How much will the strength of selection drop when ash is less common in the landscape and therefore the fungus is less common? Don't know. How many ash woodlands actually have healthy levels of natural regeneration? We had to look around quite carefully to find a woodland that had good natural regeneration. A lot of woodlands don't because of herbivores. What contribution are structural genomic variants having? We're looking into that now, but we don't know the answer yet. And also, of course, what about the emerald ash borer? That's a whole topic of its own, as we all know. Thanks to DEFRA for funding. Do look at our Ash Genome website, which we've recently revamped, and I tweet a lot. Thank you very much.
Thank you very much, Richard. And then I will now open the floor for questions. As I said before, ideally you can just raise your hand or just type your questions in the chat so that I can start moderating. I see that James Brown is really very much engaging into the discussion. Please, James, go ahead.
Sorry, I wasn't necessarily trying to get the first question again. It's just I have a point here. I mean, do we know that natural selection is happening? I think it really depends where you look, Richard. I mean, there is a wood 2 kilometers from my house. I have a wood 1 kilometer from my house, which at one time we think was the worst affected wood in the whole country, and a wood 2 kilometers from my house, where I see ash saplings between 4 and 6 meters high in their hundreds, if not thousands. I find it really, really hard to believe that an ash sapling can get to 6 meters high with not a spot on its stem and natural selection is not happening. I think really I would go with Erik that it is happening, and I think that we need to understand why. I think in the case of this wood, I think the reason is that it is a very popular place for people to walk their dogs, and so there are very few deer in there. That's my guess. It's not a rigorous observation. But going back to Richard's talk, the content, all the maths and the genomics, Richard, would you say that your conclusion that natural selection is difficult to detect, is that to do with the genetics or with the environment? I don't know Marsden Wood, but the couple of pictures you showed suggested there was a lot of variation in the local environment around each individual tree. And that's going to, I think, probably limit the extent to which you can make genomic predictions about resistance, isn't it?
Yeah, I think that was my point, really, James. The reason why our genomic predictions are not good predictions of the phenotypes we see in the wood is because of the all of the environmental variation. But natural selection is happening, and we can pick that up with the genomic predictions. And we think that's because young ash seedlings are dying very rapidly within the wood, and they were a missing fraction that were invisible to us. Because we went in at a certain time point and scored the trees and collected samples, but the fungus had already eliminated a large proportion, we think, of the seedlings in the woodland. And so natural selection is happening, even though there are these big environmental effects. So we definitely think natural selection is happening in this woodland. Yeah.
Okay. I'll tell you what would be very interesting would be to come and look at this wood near my house as an example where, you know, just looking with your own eyes says that something really big is going on. And I think there are other sites in Norfolk seeing this on not such a large scale, but I know a couple of other woods where we're seeing, you know, many square meters of recovery. And this wood is I don't know, 100 hectares of recovery. 100 acres, 40 hectares.
Oh yeah, I think there's lots of anecdotal evidence that natural selection is happening. But what we've tried to do is show at the genetic level that it really is happening, and I hope we've shown that.
Yeah.
Thank you very much. Unfortunately, we are really running out of time, and it would be nice also to see how we can build on this series of webinars and the knowledge we've been sharing, and also to keep engaging with the community that evidently is really very much willing to ask questions and to see how to collaborate to bring the ash back in our landscapes. I would like to ask Richard to conclude this webinar by sharing with us how you are confident that we can see ash coming back into our landscapes.
I think ash is not going to go extinct unless the emerald ash borer comes in and wipes out everything. If we're only dealing with Ash Dieback, I don't think it's going to go extinct. There are lots of places already with fairly low disease pressure. I think we're seeing the loss of lots of trees. I think the main question is, will it come back as a good forestry species? Will it come back as something that a forester would want to plant because it will give him an economic yield? And I think for that to happen, we do need a breeding program. I think natural selection is going to get us to a sort of equilibrium point where trees are still being damaged and are never fully resistant. But I think with a breeding program, we might be able to do better than that, and of course faster than that. And so I think the main driver for a breeding program is to get something that's commercially viable. I'd be interested if Erik has comments on that, because he's probably thought about this a lot too.
Yeah, I agree that S will stay here, and I think that we've shown that using our technology in cooperation with practitioners, we can bring ash back in the forest. I'm very optimistic about the new second generation seed orchards that we established, and I look forward to sharing the information because they look really tolerant or resistant or low susceptible or whatever. I mean, they are. It seems that there's a very strong response section. I should say that there's something about the quantics here that, besides, you know, that when we just use this breed equation, I mean, there's also a challenge to understanding the background behind that. So, but I mean, I'm also optimist, and I think we sharpened us.
Then with this, I thank you very much, and I thank also everyone for following us during today and during this series of webinars. And I wish you the nice continuation of the day. Thank you very much, and goodbye.